Here we are for the beginning of season 3. We have such a wonderful guest today. We have Ben He from Carnegie Mellon, who's going to tell us all about some work that he's been doing in rodent models. And so this is really exciting. Ben, first, thanks so much for joining us. And could you tell us about yourself?
Good afternoon. Thank you, Kim and Keith, for having me. So I am a biomedical engineer. I work on electromagnetics and recently quite a bit on acoustics. So my interest in this field actually going back many, many years ago when I was working on magnetoacoustic imaging technique, which is something we work out in our lab in order to image the tissue impedance distribution. So we inject magnetic energy, and we found that the tissue is going to emit ultrasound. So we record ultrasound signal, and we were able to actually reconstruct the tissue electroimpedance distribution at a very high spatial resolution. Now, by working on that imaging project, later on, I realized that theoretically, the reverse mode of that theory actually is the ultrasound stimulation. So with my interest in neuromodulation, I work on TDCS and TMS, and I was very happy to learn about that. The TUS, or transcranial focus ultrasound, had a very unique property, and with that technical background, and recently that this project has become one of the major research programs in my lab. So I think that is basically my background and how I got into this field, even though I started with electromagnetics, work on interplay between electromagnetics and ultrasound, but now and with a lot of interest working on ultrasound neuromodulation.
Oh, that's super. And how big a lab do you have, and how much of your lab is working on ultrasound?
So my lab currently, we have 3 research directions, all under a theme which I call systems and neuroengineering. So we work on mapping, and which is from EEG, MEG to do dynamic brain mapping. And we do brain-computer interface by using EEG through sensing and imaging. We try to decode human intention to control a device, such as fly a drone or control robotic arm. And then the third project, which currently maybe about 40% researchers in the lab are working on ultrasound neuromodulation, is we try to encode information into central nervous system. So not just doing decoding, but all of them could be integrated under the umbrella as a bidirectional brain-computer interface.
Yeah, that's great. So tell us what you've queued up here for us to listen to today.
Sure, I would be happy to share some of our experimental investigation, try to categorize of the effect of focused ultrasound and using in vivo rodent model. So this is a slide just taken from a review paper we wrote several years ago. And the neuromodulation, as most students would know, is essentially represent a neurotechnology that you inject a variety of energy into the brain or central or even peripheral nervous system. For example, left side showing invasive neuromodulation, very typical, successful DBS, deep brain stimulation, or cortical stimulation. The right-hand side here, this picture illustrates 3 most widely studied non-invasive neuromodulation techniques, starting from transcranial electrical stimulation, transcranial magnetic stimulation, and transcranial focused ultrasound neuromodulation. So this slide just to have a high-level picture to illustrate. You have an ultrasound transducer array. It's non-invasive. and it could target acoustic energy at a particular location of the brain. Now, what is special of the ultrasound neuromodulation? I think, to me, I believe that these two aspects listed here on the left side of the slide is probably make ultrasound different than other non-invasive neuromodulation. Number one is it's a high spatial specificity. As what I mentioned, my background initially came from electromagnetics. So either TDCS, TACS, or TMS, they are all electromagnetics-based neuromodulation. If it's electromagnetics-based neuromodulation, there is a volume conduction problem, which is a fundamental physics in the sense, whether you want to inject magnetic energy or electric energy into the brain, you cannot get to a focal point. That is because the field is going to be diffused. That is something, it doesn't matter how you can develop your instrumentation system. Actually, I was always amazed by colleagues working on electrical or magnetic neuromodulation field. They are very clever. I'm doing great work, but there is a limitation that is a fundamental physics, limiting the spatial specificity of any of electromagnetic-based neuromodulation technique. The second is a deep brain penetration. I think ultrasound, which can get signal into the deep brain. So we will get to that later by showing some experimental results. So I try to make my point by sharing 3 experiments, all in rodent model. The first experiment is, as illustrated in this slide, you can see that in the cortex, there's a variety of this cell type. For example, excitatory neuron showing in the top, interneuron. So with different kind of morphology configuration. So the first question is, since we talk about ultrasound neuromodulation has high spatial specificity. So the question that my colleague and I have been asking in the past several years, are we able to selectively target at a certain neuron type? For example, target at excitatory neurons or target at interneurons? So this is one question that we did some experiment. I wish to show this experimental result first. So another is there's a question, what is the mechanism of the ultrasound neuromodulation? There's a lots of hypotheses or theories out there. And so these are certainly could be considered mechanosensitive ion channel hypothesis with the recent Caltech publication at the Nature Communication. I think that hypothesis is receiving a lot of experimental support about this mechanism, but there are some others like membrane capacitance, pseudoporation, membrane wave thermal effect, and so on. So there are certain different groups that have different view, even though, for my group, and we share the view that the mechanosensitive ion channel might be a possible mechanism make the ultrasound be able to activate or inhibit neuronal activation. So but this could be illustrated from here, the acoustic radiation force. From the physics, this could be represented by something that proportional to a important parameter of the ultrasound, so-called the pulse repetition frequency. So in the sense that the pulse repetition frequency, which is illustrated here, that is proportional to acoustic radiation force. So one of the thinking we had is if we would change this pulse repetition frequency, we can control the acoustic radiation force And if that ARF is a major driving force for the mechanosensitive ion channel mechanism, you can control the neural excitation or inhibition. So this is the first question that we try to address by experimentation. So
Wait, could I ask you some questions before we move on?
Yeah, sure.
So you've very nicely listed a number of possible hypotheses here, but they're not necessarily exclusive. Like you could have several of these working together. You could have membrane waves working on mechanical sensitive ion channels, for example. Do you see it that way, or do you see these as more exclusive?
Kim, you're absolutely right. I don't think they are exclusive from each other. I mean, there certainly is this different angle, and at this time, to my knowledge, I don't think we are very clear about the mechanism yet. So more research would be needed, in my opinion, to know exactly, just like we know how neural excitation from the electrical process, which is very, very clear. But it's not entirely clear. I think there are some work, and this mechanosensitive ion channels work is a recent experiment, is very interesting and persuasive to me, but I think there are multiple possible mechanisms. It could happen at the same time, as you said.
Yeah. So I have another question here on your equation down here at the bottom, because I think it's maybe a little counterintuitive to think that the radiation force is related to the temporal average of the intensity. It's because people often think of the temporal average as being sort of related to the ability to heat the tissue, which is a little bit different than sort of the ability to distort the tissue, which maybe is related more to the peak intensity. So I'm curious as to your thought as to the PRF playing such a role here with respect to mechanical sensitive ion channels.
It's not our work. This is what's out there. Are you suggesting that this equation?
No. I mean, the equation's fine. So the intensity is pressure squared over the acoustic impedance, and then when you talk about the temporal average, then you take into account the duty cycle. So actually, I think you might have duty cycle there, instead of well, but anyway. But usually the radiation force. I mean, the way I think about it is radiation force is sort of like the force on the tissue, sort of giving a mechanical distortion of the tissue, a compression or expansion of the tissue, which isn't necessarily related to the ability to heat the tissue. But on the other hand, the temporal average could actually play a role as well. So for example, if you say it has to have a certain distortion over a certain amount of time, then temporal average maybe plays a role. So I'm not saying it's wrong by any sense. I'm just like, oh, it's so interesting. And I'm wondering what your perspective is, because in some ways, I guess, it sort of takes into account the intensity times time. So it is sort of, for me, like the area under the curve maybe is related to the temporal average. So actually, no, I just find it really interesting. I don't mean to like sidetrack us at all. Why don't we keep going and see where you're going with this?
All right. So now the question would be, what would be the effect of this PRF and acting on the neuronal cell type selectivity or different response? So we did experimentation. In vivo rat, we inject focused ultrasound energy to somatosensory cortex, and then we use a multi-electrode array. So we record the signal, and then by selecting the single unit and the response, and it's well established in neuroscience that different kind of shape of the action potential based upon the initial phase or after hyperpolarization, and we could quantify or categorize these neurons into the regular spiking unit or presumably excitatory cells, or fast spiking unit or presumably the inhibitory cells. Now, here is a and here's ultrasound stimulation. This yellow tiny guy. And you can see for excitatory cells, we found that there's very clear response once the ultrasound stimulate, and in these excitatory cells, their normalized this neurospiking rate is going to increase. If you're having higher PRF, here is 300 hertz. Here, if you go to 3000 hertz, then you can see the spiking rate is increased proportionally. Now, which was anticipated intuitively, but what is interesting is for inhibitory cell or the fast spiking unit, there is no noticeable that spiking rate corresponding to external ultrasound stimulation at the same location. So
wait, can you go back? So it looks as if you're saying that there's no response at all from the fast spiking.
we did not, I mean, there's some fluctuation, but we did not observe statistically significant response. Maybe the next slide would see better, because here you can see this is an inhibitory neuron. We change our PRF from 30 hertz, 300 hertz, 1500, 3000, 4500. So 5 different PRF, and we do statistical analysis, and there is no statistically significant difference. By changing PRF, their spiking rate does not change. On the other hand, for excitatory neuron, you can see that the lower PRF, usually you have less spiking rate, but by increasing PRF, it will gradually increase, and there is statistically significant difference or increase in spiking rate by increasing the pulse repetition frequency. So this is where we came from. We are still, this is a phenomenon. We're not saying it's a mechanism, but that if PRF goes up, acoustic radiation force goes up. I mean, there may be some minor difference. That if the mechanosensitive ion channel mechanism hypothesis, if it would hold, this is one possible way to explain this correlation. Kim, please.
And for these experiments, did you change the pressure or the peak intensity? Because as or did all of these have the same? Did they have the same temporal average? Yeah,
this is a very good question. So the effect of the intensity, right? And if you change that, so for our experiment, this is something I mean, it's based upon translational needs. I mean, we're not just studying experimental animal. We want to eventually go to human translation. So we are intentionally keep the pressure low. Certainly, if you actually change that pressure as a parameter, there will be different. There may be something different, but here we are using as the lowest possible intensity. So hopefully that could be translated to human application, so you don't have potential safety concern in humans.
Yeah, that's super. But I'm just confused. So if we use the same pressure for each of these pulse repetition frequencies, then as you go to the right, is it just a question of using the same pulses, but sort of squishing many, many more of them per second, so that you have more of the same amplitude pulses every second? So
that's a very good question. We did look into effect of duty cycle. Mm-hmm. So we did not change the intensity parameter. So I don't have that figure with me here, but in the control experiment, while we changed the duty cycle, we did not observe similar phenomena. Mm-hmm. So we keep so in that way is certainly because duty cycle is another parameter which may impact the acoustic radiation force, as you pointed out earlier, right? But in this case, we found that this parameter appears to be sensitive. We're not saying this is the only parameter, but it seems to us that this PRF is one of such important parameter, at least that is experimentally exhibiting such kind of cell type distinctive response.
Mm-hmm. And do you remember what was the pressure that you used for these?
It's in the paper. I don't remember exactly, but if I recall correctly, we use the pressure around the range of 130 kilopascal.
Okay, great. Mm-hmm.
And for specific, I apologize, don't remember exactly. This is in the documented publication, but that's about the range. Or sometimes we also use experiment do 99 kilopascal. So basically about 100 kilopascal.
Yeah, okay.
So we did another experiment to verify in the beginning. After we get this result in rat, we were very excited, but we are not so sure if that's true. So we did optogenetic transgenic mice model to confirm in this particular case. So using optogenetics, we got this PV neuron and CAMK2 alpha neuron. And we show very clearly there's the difference between these two neuron type in terms of initial phase and after hypopolarization. So you can see that this red color is PV neuron, and the blue color is CAMK2 alpha neuron. So from there, and we tested in the PRF 30 hertz and 300 hertz. And what is interesting is the CAMK2 alpha neuron show the similar trend that the lower the PRF, then the smaller the spiking rate, and the increased PRF, you observe the increased spiking rate. A similar phenomenon that we observed in the in vivo rat model for excitatory neurons. In this case, what is interesting for the PV neuron is it's not like in rat, it's insensitive. In this case, there's an inverse relationship. You increase PRF, but then you see the decrease. But at least they two different distinctive response to ultrasound stimulation for these two neuron type. So now, what causes this? I mean, to be honest, we are not very clear. We just have some, interpretation, but the mechanism is still not very clear. And here, this is a cartoon to illustrate that for regular spiking unit, you can see the radiation force and the change and lead to more ion channel response. And then this faster spiking unit, but this radiation, acoustic radiation force does not play a role. So this could due to the different type of ion channels and different cell type. And also it could be, as what we discussed earlier, that the shape, the surface area of the cell membrane and arrangement configuration in terms of where the ultrasound energy comes, it may be that is a difference, so show different behavior. Or the third possible reason could be due to glial cell, clearly not neurons. But anyway, these are all some possible thinkings, but we are not very clear about what caused the cell type specific response. But we does know is these data illustrate that excitatory inhibitory neurons in rats somatosensory cortex and show intrinsically different in response to ultrasound and the pulse repetition frequency as modulated by PRF. And this result suggests that we can target a specific neuron type non-invasively, so which is in the positive tone, even though in the negative side, we do not know what exactly happening, but the positive side is we did observe this experimental finding that non-invasively, by tuning ultrasound parameter, you can target at a specific cell type. So for that, we were quite happy to see these experimental results because that may have important translational potential for human use.
Mhm. Yeah, I think there's a lot of data that says as you have kind of more duration and or more intensity that you can get more response from your excitatory neurons. I've sort of questioned still, or I'm trying to wrap my head around why splitting it up into lots of little pulses is meaningful on the neuronal level. Do you have any insight into that?
So, Kim, is your question why use a pulses of pulse or?
Yeah, it's, the general question of a pulse versus continuous wave. If you think of like a continuous wave as having a pulse that's a certain amplitude and duration, and then you split it up into lots of little ones, why does splitting it up, like, what's your thought about the neuron, how it responds to lots of little ones as opposed to a longer single pulse?
That's a great question. I mean, I could tell you that that's my puzzle. I was asking myself why that PRF is important. Now, the reason we are splitting into continuous wave into many, many little one is because we do not want to have possible heat buildup, which might be the case for continuous wave. So you need to split into many, many little one in that way to control the temperature rise. I'm certain that's not the only reason, but this is important consideration in my understanding. Now, what we do see is because once you split into many, many little one, and that pulse repetition frequency, so I mean, certainly that is a phenomenon. I mean, what it would take is could it be, Kim, as you mentioned, is it's not how you split it. It could be the accumulation of all these many, many small ones that play a role. But we do not know what exactly is the single driving factor. So we observe the phenomenon, but we don't have a clear answer for what's causing this phenomenon at this time. Okay, I guess I should speed up and quickly. So the next I want to quickly show the two other experiments. One is a long-term effect. We talk about human translation. So there's one question is, you have ultrasound stimulation, you see some effect. Now, what about if the ultrasound stops? Will this effect still lasting, or it will be gone? Because if you want to use it for therapeutic use, you do want to see this lasting effect. So what we use is we look into this long-term potentiation or long-term depression model, which is well established in neuroscience from electrical stimulation. In this case is we do electrical stimulation and as a baseline, and after that, we do 5 minutes ultrasound stimulation or control and compare with electrical stimulation, and then follow up by 30 to 60 minutes and electrical recording to see what would be the effect. So very quickly is we are very surprised and interestingly to observe that our data show no long-term potentiation phenomena. However, our data show very clear long-term depression phenomena. So you can see here is the slope, which is measured in the FEPSP during this phase, and there's a clear that the change decrease in this slope. And this phenomena we compare with, this is a focused ultrasound, this color, and the blue color show if it's a conventional electrical stimulation. And what we found is for 24 minutes, the ultrasound exhibit similar long-term depression effect. And after 24 minutes, this essentially returning back to baseline. And basically there's a big difference from this LTD caused by electrical stimulation versus that caused by the ultrasound. And in this case, we use a relatively high PRF from 3 to 10 kilohertz. And during this range, we did not see statistically significant difference in terms of how this PRF is going to impacting this EPSP slope. So this is another experimental finding. We saw that in hippocampus in rat, for 5 minutes ultrasound stimulation can lead to up to 24 minutes long-term depression. So which is a positive indication about a potential future application, not currently, that if you stimulate, if you have lasting effect, which is really what we need for human neuromodulation application.
Mm-hmm. Why is it then that electrical stimulation has a longer term effect than ultrasound?
Yes, that's right. That's to my understanding, we record like 1 hour, it will last to the 1 hour. So electrical has a longer, but ultrasound for our experiment, that effect stopped after 24 minutes. Mm-hmm. But on the other hand, it's only 5 minutes ultrasound. So we didn't test the longer duration of ultrasound stimulation.
What's the mechanism by which electrical stimulation has has an effect over long term?
Well, this is a well-established neuroscience finding that in this in hippocampus, it was found that if electric stimulation, you can see LTP and LTD both. And what is we are puzzled in the beginning. We didn't observe LTP, but only LTD. On the positive side is this LTD phenomenon, and the 5 minutes ultrasound stimulation can lead to almost 30 minutes lasting effect. Now, why cause that? That will be I think will be the neuroplasticity and occur in the central nervous system, which is established previously. Okay, so the next few minutes, I know we are probably running late, and I want to very quickly show some of our latest observation. Once again, it's experimental investigation, but more try to moving towards translational aspect. So this is to use cranial focus ultrasound for pain management. And particularly, we look into sickle cell disease model, and there's sickle cell disease is a well-established, the red blood cell with the sickle shape. So it causes pain everywhere in the body, and particularly impacting African American. So what did we do is we use in collaboration with the Kalpana Gupta's group at the University of California, Irvine, and she has a SCD mice model. So we do the mice experimentation. We do behavior assessment before ultrasound treatment, and we have ultrasound treatment. We have sham control, basically directing ultrasound to a different location over the skull in the brain. And then we have negative control, that is, do not emit any of the ultrasound energy. And after stimulation, and we do another assessment, behavior assessment, to see what is the effect on pain sensitivity. So we try in the wild type of mice first, and you can see that it's 3 minutes after stimulation or 8 minutes after stimulation in both the male and female and the mice. We see statistically decrease in the pain sensitivity following ultrasound stimulation. And in the sickle mice, and what is in this case, what is interesting, there's one phenomenon, just like what we observe, different distinctive response in the somatosensory cortex in rat, that the excitatory and inhibitory cells has a different response. In this case, the red color show if we use a low PRF, the green color show if we use high PRF. Actually, the low PRF is going to decrease pain sensitivity in the sickle cell mice, but high PRF increases the pain sensitivity in the sickle cell mice, and 3 and 8 minutes post-stimulation. We also stimulate at insulin, and we found in this case up to 30 minutes post-stimulation, we found the low intensity and the PRF can decrease the pain sensitivity. And in this case, actually the high PRF does not increase sensitivity. So it looks like there's a different brain region, the performance is different as well. But in short, this is what we found is ultrasound stimulation, a single session can decrease the pain sensitivity in general, but for somatosensory cortex and the different PRF seems to have different and the performance. And finally is in the insular stimulation up to 30 minutes post-stimulation, we observe that decreased pain sensitivity following ultrasound stimulation. So to wrap up, I want to acknowledge all the people who made a contribution to this research while I'm presenting here, and including my faculty collaborators and student postdoc and in NIH funding support. And thank you. And thank you, Kim and Keith.
Well, thank you so much. Maybe we have just a couple more questions. So with that last one, when you were decreasing pain in your sickle cell anemia mice, where were you targeting?
We targeted multiple nodes, and in the data I presented, we targeted somatosensory cortex, and we targeted at insula. And these are the nodes we learned from a previous research project that I was doing is using neuroimaging, EEG imaging, and a functional MRI in sickle cell disease patient. And then by doing neuroimaging, we found these nodes are where seems to be involved with the sickle cell pain. So we are now just like a stimulate at this target in the hope is going to decrease the pain sensitivity. And this mice study seems to support this notion.
Ben, we love your approach to cell type specificity and monitoring that sort of thing. Are there any avenues within pain sensitization where one cell type might be more important for your treatment than others? And are you going to try to merge these projects together down the line? We're excited to see more in that avenue. So we're rooting for you.
Wow, Keith, I mean, yes, if we would understand everything and why all these happen, right, we can design a rational or optimal way to treat disease. So this come back to my early observation in the beginning is I think focused ultrasound neuromodulation is a really very exciting emerging technique. But as what we discussed, Kim raised a lot of questions. I'm like, why we're going to observe the cell type specific response? There are many unknowns. So I don't think that we know a lot of them. So my observation is at this time seems various labs are using different experimental approach to try to approach this problem from different angle. At a certain stage, I think we would be in a position to design a rational optimal approach, but maybe at this time, variety of investigation is very much needed. We need all approach from all species, all the techniques. I mean, that is my observation. I mean, sometimes it's just too new.
Yeah, it's a little daunting. I think even in the other fields like TMS, where you might see low or high pass filtering by certain cell types, even then there's just too many channels and too many membrane properties to start to get into that. I mean, of course we'll get there eventually, but I appreciate your thoughts on that.
Yeah, absolutely. I think you're right. Even in other neuromodulation like a TDCS or TMS, also there are many parameters associated with this neuromodulation. But with the ultrasound particularly, I think the number of parameter is my experience is much more than other modality. It's just because like the Kim was a common, this little guy, there are many little guys. So you have much more dimensionality compared with what you can play with, even in TMS. But regardless, I think it's a lot of fun. It's new and it's exciting. So eventually, I think we're going to find out exactly what's happening, so we could say, yeah, because of what, and so we are doing this. But now is, I think the data we found in from my lab is just adding to some of the knowledge that the field and the community is working on. And the Keith, I agree with you. And we are particularly excited by this cell type distinctive response ultrasound exhibit. I think that is experimental evidence indicating the strong potential and for non-invasive human neuromodulation application.
Any last questions, Keith, before we let Ben off the hook here?
I'm
Let him
good.
go.
We'll get some offline questions.
All right. Thank you so much, Ben.
Thank you, Ben.
Well, thank you. It's a pleasure. And thank you for running this. This is great. Have a good one. Take care.